Good morning, good afternoon, or good evening, ladies and gents. Welcome to Biomed. I hope you guys all had a good weekend. Today we're going to take some notes on isolating and identifying bacteria using the aseptic technique. We're not going to go through it all today, mainly because, again, I want to respect your time. So let's hop into it. A couple things that you do need to know is the differences between bacteria and viruses. Viruses are not considered to be living cells. They are not alive and they are not dead. They're neither. They're not eukaryotes and they're not prokaryotes. They're considered their own separate family, if you want to. If you ever go to the doctor and say you have a viral infection and he gives you an antibiotic, girl, you got to run. That antibiotic isn't going to help you at all, and that doctor probably isn't a doctor. So you should never be giving antibiotics for a viral infection. Kind of a neat thing is once you get a viral infection, you will always have that virus inside you. So if you ever had chickenpox, that is a form of herpes virus, you will always have that virus inside you. That doesn't mean you'll never get it again. Um, you can always catch the virus again, but generally speaking, it won't be as strong as the first time you had to deal with it. So some of you might have had colds before. You can get a cold again, um, but it typically isn't going to be as strong as the first time that you had. Viruses also are considered to be considerably smaller than bacteria. Think about it in a couple different ways. Viruses infect cells, and bacteria are one individual cell. So the virus actually has to go inside that bacteria. So viruses are much, much smaller than bacteria. Bacteria themselves are one-celled organisms. If you ever get infected with a bacteria, you get sick with a bacteria, like a sinus infection, typically is a bacteria infection, not always. And you go to the doctor, they'll give you a dose of, or they'll give you some antibiotics. It's extremely important that you take the entire dose of antibiotics. You don't stop as soon as you start feeling better. This helps get rid of the bacteria that's in your system. Again, you can get that same bacteria again later on and still get just as sick. And then in that case, you need to go back in for antibiotics again. The link below brings you to an anatomy website. You need to enable Flash. Remember, click on the giant lock or up by the URL bar and enable Flash. You'll be quizzed on the anatomy of bacteria, so make sure that you do this. You don't have to send me a picture or anything, but just make sure that you practice this, because again, you'll have a quiz on this content probably tomorrow on the anatomy of bacterial cells. All right, here's a picture of a bacterial cell. We're going to go over a couple of the different anatomical features, and again, you do need to know this. The first one we're going to talk about is the flagella or flagellum. It kind of looks like a tail if you want to look at it that way. It is used by the bacteria to move. So in a liquid system, like in water or inside your body, this is how the bacteria gets around. The second thing that we're going to talk about, number 2, is the pili or fimbriae. Fimbriae basically looks like and sounds like fingers, and that's what they do. The bacteria uses it as like a finger. It's used to latch on to a host cell, or to like the intestinal lining inside you, or to other bacteria in order to reproduce. Number 3 is a nucleoid. Bacteria are considered to be prokaryotes, so they do not have a nucleus. So the nucleoid is basically a long strand of DNA for the bacteria, and this is how the bacteria can reproduce asexually, is with this strand or with this nucleoid. Number 4 is the plasma membrane. Functions just like our plasma membrane does, and it basically keeps the bad things out and lets the good things in. Number 5, the ribosomes. Can you guess what that makes? Yeah, it makes proteins. I know it says on the screen, but So number 6, the cell wall. Very similar to a plant. It is very stiff, very rigid. Again, protects the bacteria from any kind of infections or anything like that. And yes, bacteria themselves can get infected. Anybody want to take a guess at What infects a bacterial cell? If you were thinking viruses, you would be correct. Well done. Number 7, the capsule. So this is the outermost layer of the bacterial cell. So it goes capsule, outermost, cell wall, second layer, plasma membrane, inner layer. The capsule, generally speaking, for most bacterial cells, is very sticky. This allows it to, again, latch onto different surfaces. So if you sneeze, the bacteria floats through the air and then it can latch onto or stick to your skin, like your hand. And then when you bring your hand up to your face to touch your face or to eat, and then the bacteria transmits itself into your mouth, the nose, or your eyes, and that's again how you get sick. And lastly, number 8 is the plasmid. So this is a small fragment of DNA. This is kind of like the cool part of bacteria, I think. The plasmid is used in sexual reproduction for bacteria. So the bacteria can both reproduce sexually or asexually. And this is the plasmid. If the bacteria reproduces sexually, this is what's transmitted to its partner. This is also something scientists use to their advantage. We can take the plasmid out of a bacterial cell and put whatever DNA we want inside that plasmid. So when that bacteria reproduces, it passes that DNA on to the next generation of bacteria, and bacteria reproduce very, very quickly. So that could be a cure for different genetic diseases and things like that. An example of scientists using plasmids and bacteria for our advantage is we've been able to create the protein that makes insulin. So we take the DNA out of the bacterial cell, put that protein into that plasmid instead, and then every bacteria from that generation forward starts to create insulin, and we can harvest that insulin for diabetic patients like Anna Garcia. Some more things you need to know about bacteria are their shapes. They typically come in 3 different shapes: spherical, rod-like, and spiral. The spherical bacteria are going to be coccus bacteria. The rod-like are going to be bacillus, and the spiral will be our spirillum. Some examples of them are, some of the examples again of the coccus bacteria are going to be staphylococcus. So if you ever get what's called a staph infection, that type of bacteria that has infected you is in the coccus shape or spherical shape. An example of a bacillus bacteria is Bacillus anthracis, which creates anthrax, which I'm sure most of you have heard can be extremely lethal to humans. And finally, an example of a spirillum bacteria is Treponema cuneiculi. I might have mispronounced that, but that is syphilis that can be found in rabbits. And now on to viruses. Viruses typically come in many different shapes, but there's really 2 different types of viruses. There's bacteriophages and then viruses like influenza. Bacteriophages attack bacteria. So what they have is, well, all types of viruses are protein coats, which align the outer parts of the virus. With inside that protein coat, it's going to be a nucleic acid with the DNA, or with a strand of DNA. In the case of a bacteriophage, it's going to have a tail and fibers. The tail, for the most part, is kind of like just a long cylinder column. You really don't need to know much more than that. And the fibers kind of work like arms and hands. So the bacteriophage will float through your body, and when it comes across a bacteria, whether it's a good bacteria or bad bacteria, those fibers will latch on, and then the bacteriophage itself will insert its genetic material into the bacteria, and then will use that bacteria as a host to reproduce. The influenza on the right-hand side basically does the same thing, except for it floats around, and you can kind of see these little hairs on the outside. Those will connect to specific molecules on your cells, depending on the virus and depending on the cell type. And once they have a match, they'll stick together, and then the virus will insert its genetic material into that cell and then hijack that cell for its own devious purposes, which in other words basically just means they're going to make more virus cells over and over and over again until the original cell that it infected explodes, releasing more viral components. All right, and that wraps up what we're going to talk about for notes today. Make sure you check Schoology for the Amoeba Sisters Ed puzzle. It's only like 4 to 5 to 6 minutes long. All this information that was on here today is potential test material or quiz material. I hope I was a good host for you guys today. Ha 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 ha. And that these notes have infected you and that your learning is very infectious. Okay, I'm struggling now. Just being real with you guys. Have a good rest of the day. Make sure you guys get this in, and we'll be back tomorrow.